You might have one in your pocket right now, or maybe you see someone holding one every single day. Genuinely, globally, almost universally, we've all seen one. And if you are sitting somewhere watching this, it's highly likely that you or someone you know will light one up and inhale it today. Maybe you even consider it a harmless habit. But here's something that nobody tells you. Under the right conditions, that single drag is making things worse. Not a little worse, potentially fatally worse. Inhaling that smoke doesn't just deposit black tar in your lung tissue. It halts your cellular defense system. It triggers a genetic process, and that will kill you. And right now, in bodies all over the world, those conditions are created every single day. This is the story of the p53 genetic shutdown, a cellular mutation process most people have never even heard of. The threshold a human cell cannot negotiate with, and a daily hazard that sits closer to your face than almost anything else in your life. But to understand why, you first need to understand what your body is actually doing when you breathe. And to understand why a cigarette can kill you, you first need to understand how your cells actually protect themselves. Because most people think cigarettes only kill you when you're old or a heavy smoker. They don't, not exactly. Mutation is the inevitable tax of cellular life. Every single day, your cells divide to keep you alive. But during this copying process, some errors are inevitably made. Under normal conditions, your body handles this by actively identifying and repairing those genetic errors. There are no manual overrides inside your DNA. It relies entirely on a master protective protein called p53 to identify and destroy mutated cells. Without a functioning p53 gene, cellular repair is zero. It does nothing. This is why breathing normal air feels completely fine, while inhaling cigarette smoke is incredibly dangerous. In a clean environment, your cells' repair mechanisms work freely around your DNA, fixing errors. In a smoke-filled lung, on the other hand, a thick layer of sticky tar acts like a physical barrier, trapping toxic chemicals and keeping them pressed directly against your cellular lining. Your body produces mutated cells constantly, just from the physics of living. At normal baseline exposure, it's managing a tiny, harmless fraction of genetic glitches. Put a burning cigarette to your lips, and that number climbs fast. A single puff introduces over 7,000 chemicals, including 70 known carcinogens. And all of that toxic load has to go somewhere. Cellular repair is the primary defense route. And when that defense route is overwhelmed, something has to give. And scientists and geneticists found a way to measure exactly when that defensive system starts to fail. It is called the p53 mutational hotspot. Most people have never even heard of it. Cancer biologists have been mapping this problem for decades using molecular gene sequencing. Inside a human cell, your DNA replicates using a sequence of billions of base pairs. When things are healthy, the replication process is stable and predictable. The cell functions perfectly. In a clean lung, the cellular DNA remains completely safe from critical mutations because the p53 protein remains active and the cell repairs itself. Under a layer of toxic tar, that gap closes. The genetic damage creeps toward a critical threshold. And if the protective gene itself is mutated, there's no cell repair at all. The cellular mutation rate and the tissue damage begin to feed off each other. And this mechanism, the p53 gene inactivation, is arguably one of the most critical biological thresholds that most people have never even considered, because it doesn't just tell you how damaged a cell is. It tells you how close the chemistry inside your body is to spiraling completely out of control. It tells you whether your body's built-in defense system can protect you or not. And there is a specific biological coordinate beyond which your cellular chemistry simply cannot cope. So what is that threshold, and what does it actually mean for your body? Codons 157, 248, and 273, Those are the coordinates. Beyond them, the biology stops working in your favor. A standard human cell replicates safely under normal repair conditions. But in the presence of a chemical in cigarette smoke called benzo-A-pyrene, a highly toxic metabolite called BPDE is formed. This metabolite bypasses other parts of your DNA and homes in directly on those specific codons on your p53 gene. It binds covalently to the guanine bases. Your cell keeps trying to divide as it sustains life, but there is no longer any protective protein to stop the errors. The genetic errors begin to compound, and unlike a normal cell cycle where the body might safely pause the process to fix the DNA, this growth is uncontrolled. The damaged cell triggers more divisions, which carry the mutated gene, which triggers even faster, uncontrolled replication, cellular runaway, genetic venting, tumor formation, and eventually a devastating cancer. And this is where the tar and carbon monoxide come in. At normal conditions, your lungs' natural defense systems sweep away particles. That is the whole design of respiratory clearance. But when you inhale tobacco smoke, the hot, sticky tar stops passing through and starts doing something else. It acts as a heavy-duty physical barrier,
paralyzing the cilia. The toxins your body needs to clear cannot escape. They are trapped directly against the lung membrane. The tar is now a physical trap cooking your tissues. Add a high concentration of carbon monoxide to that, and you have a worst-case scenario. Oxygen transport is already failing. Carbon monoxide binds to your hemoglobin over 200 times stronger than oxygen, starving your heart and brain. The physical barrier of tar cannot be bypassed, and the constant inhalation increases the rate at which the cellular damage builds up. Under those conditions, your body's cellular safety protocols, which usually force damaged cells to commit suicide, cannot save you. The physical genetic damage is building up faster than your repair enzymes can react, accelerating the biological process that will turn a single cell malignant. This is not a theoretical concern. This is documented. Pathologists and oncologists around the world regularly observe this cellular destruction under microscopes every single day. Hospitals around the world regularly register diagnoses, aggressive tumors, and fatalities caused by the cellular mutations written during brief, everyday smoke breaks. Now here's the part that should genuinely alarm you. Many people believe that their bodies are resilient enough to prevent this from ever happening. They think eating healthy or occasional exercise makes cancer a risk only for long-term heavy smokers. They are wrong. Analysis of genetic damage shows that when a single cigarette is smoked, the chemical adducts can bind to your p53 gene and initiate the mutation in a matter of hours. Once that critical codon threshold is mutated, the cell enters a state where unchecked division is locked into its future. The microscopic growth rate of the tumor can remain hidden for years, dividing silently in the dark. In clinics worldwide, millions of patient files tell a tragically quiet story. Patients diagnosed with advanced stage lung cancer who never felt a single symptom until the disease had already spread. It happened quietly, right inside their chest. And these mutations happen more frequently than anyone predicts, especially as lifestyle stresses increase and modern cigarettes are engineered to deliver nicotine faster. But here's the catch. A perfectly healthy, pristine, uncompromised genome is the extreme optimistic scenario. Real life isn't that. Data shows that for most individuals, accounting for aging, background pollution, passive secondhand smoke, or simple genetic variations, dangerous cellular instability begins at a much lower threshold. For an aging body that has gone through decades of life, its genetic repair efficiency is much lower, meaning it takes far fewer chemical insults to trigger a permanent error. So the threshold where it starts losing the battle against mutation arrives much sooner. This matters enormously when you look at which bodies are at risk. It is not pristine, genetically flawless individuals living in isolated, toxin-free environments. It is average human bodies with normal cumulative cellular wear. It is social smokers, light smokers, and anyone who exposes their lungs to the highly concentrated combustion of tobacco, which is exactly how millions of silent cellular fires are ignited inside human chests every single year. So, back to the cigarette. The reason that smoking can kill you isn't just because tobacco is inherently a poison. It's because the conditions in which our body's defense systems stop working and start failing us are conditions we willingly create in our lungs every single day. Conditions that are completely normalized in society, and conditions that most people have no framework for understanding, because almost nobody talks about G to T transversions or P53 gene methylation. We talk about smoker's cough. We talk about yellow teeth. We talk about lung capacity. But the actual biological limit, the genetic integrity that determines whether your cells can safely replicate, rarely makes the daily news. Codons 157, 248, 273, that is the wall. Below it, your cells can cope. Above it, no amount of organic food, no exercise routine, nothing changes the outcome. The biochemistry has closed the door. And the honest and uncomfortable truth is this: many of us are inviting that danger right into our bodies, inches from our vital organs, with no way to stop a genetic fire once it begins. Next time you reach for that pack and pull out a cigarette, now you know what your cells can and cannot do, and why sometimes the most comforting habit is the one thing that makes you deadly.